Okay, so for this class, we're going to just quickly recap the citric acid cycle and talk about two related processes, the anapleretic sequences as well as the glycosylate cycle. Just as a bit of a recap, to summarize the last class, we looked at obviously the citric acid cycle. Shown over here is just a schematic giving us some context as to where the citric acid cycle lies in the context of general glucose metabolism. So shown here is actually the complete metabolism of glucose in a schematic form, showing the breakdown of glucose, 2-pyruvate, 2-acetyl-CoA, and the citric acid cycle. Over here in the middle is the energy yield from each of the various processes in terms of both reducing agents, or rather reducing equivalents, as well as ATP. Note that these reducing equivalents are eventually converted to ATP via the electron transport chain. We want to talk about that in the next section. As you can see here, glycolysis and pyruvate dehydrogenase are responsible for around 12 ATP per glucose molecules. However, the citric acid cycle is responsible for a grand total of 20 to 22 ATP molecules per glucose molecules. In other words, the citric acid cycle is the main powerhouse of the cell and is responsible for liberating the majority of the energy stored in glucose. Whereas again, glycolysis and your pyruvate dehydrogenase release some energy, but a lot of the energy still remains. The citric acid cycle is able to generate all this energy by facilitating the complete oxidation of our acetyl-CoA acetyl group to carbon dioxide. This is an extremely exothermic reaction, and it results in the complete oxidation of your carbon to CO2, which is energetically favorable. This process contains a total of 8 different steps, which we summarized in the previous class. Just to give you a rundown of the chemical logic of this, these steps can be broadly divided into several different categories. First is we couple our acetyl-CoA 2-carbon group to oxaloacetate, which has 4 carbons, to give us a 6-carbon intermediate. We then decarboxylate or pull off 2 of these carbons as carbon dioxides. This gives the bulk of the lion's share of our energy and generates a 2-carbon species. Finally, we take our 2-carbon species and use that to regenerate our oxaloacetate intermediate. Hence the name citric acid cycle, because the oxaloacetate is coupled with the acetyl-CoA, goes through the decarboxylation, and ultimately is regenerated at the other end. Hence the need for a complete cycle. This slide here is a brief summary describing the regulation of the citric acid cycle. This is not something which I will test you on extensively, but it's something which you should be aware of. Shown here on the left is a schematic of the citric acid cycle showing the various reactions. On the right here is a summary of the regulatory pathways or the regulation of the citric acid cycle. The main points at which the citric acid cycle is regulated is the first reaction, as well as reactions The citric acid cycle is regulated at 3.5 steps. The main regulatory step of the citric acid cycle is actually pyruvate dehydrogenase. This is not technically part of the citric acid cycle, hence why I call it half a step. But again, pyruvate dehydrogenase is the main step at which the citric acid cycle is regulated. The citric acid cycle can alternatively be regulated at steps 1, 3, and 4. If we take a look over here, we can see why this is. If we look at the delta G over here on the left are the delta Gs of the various reactions. Again, the pyruvate dehydrogenase, the generation of our acetyl-CoA, is the main regulatory step. Accordingly, it has a large negative delta G. Conversely, steps 1, 3, and 4 have a large negative delta G. These steps are relatively easy to remember because they are characterized by, again, this large negative delta G. In the case of citrate synthase, this large negative delta G comes from the fact that you're cleaving this thioester bond. This thioester bond is extremely energetically unfavorable. Cleavage of this bond is very energetically favorable. So that accounts for why step 1 has a large negative delta G and is a main target for regulation. Steps 3 and 4 have a large negative delta G because these steps result in the release of carbon dioxide. Again, the release of carbon dioxide is extremely energetically favorable, giving rise to your large negative delta G. Steps 5, 6, and 8 result in the release of high energy carriers such as GTP or our reducing equivalent. It's very tempting to think of these as potential regulatory steps because they result in the release of energy. However, the delta Gs for these reactions is actually relatively small. So even though you're getting some high energy components out of these steps, they're not actually good targets for regulation just because the delta G is close to 0. So thus far we've talked about the citric acid cycle in the context of a catabolic process. In other words, we're taking acetyl-CoA, we're taking carbohydrates, and we're breaking them down to generate carbon dioxide and energy. However, it is possible to think of or view the citric acid cycle as an anabolic process. This is because many of the intermediates from the citric acid cycle are actually important precursors for various biosynthetic reactions. In other words, these are anabolic processes. This is where the anaplerotic sequences come in. The anaplerotic sequences are a series of pathways or a series of reactions through which various citric acid cycle intermediates can be removed to participate in biosynthetic processes. Conversely, the anaplerotic sequences allow us to replenish our various citric acid cycle intermediates, which have been removed in such a fashion. And we will talk a bit further about how that works, or we'll talk a bit further about how that works in subsequent slides. For now, though, I'd like to introduce you to the three main categories of anaplerotic processes or anaplerotic sequences. The first is the removal of citric acid cycle intermediates to form carbohydrates. These are summarized in these red and black arrows shown over here on our schematic and illustrate exactly which intermediates can be pulled out and removed. As you can see here, oxaloacetate can be interconverted. As you can see here, oxaloacetate can be pulled out of the citric acid cycle to participate in gluconeogenesis. This is something which we saw before when looking at gluconeogenesis. Conversely, pyruvate can be directly converted to oxaloacetate, again something which we've seen into oxaloacetate. We saw these reactions in the gluconeogenesis pathway and in the context of gluconeogenesis. However, via these same mechanisms, oxaloacetate can be taken directly from the citric acid cycle and used in the biosynthesis of glucose. Again, taking an intermediate from the citric acid cycle and using it for an anabolic process. Conversely, malate over here can also be converted directly into pyruvate through the malic enzyme. This provides a second route through which the intermediates of our citric acid cycle can be used as precursors for biosynthesis. Conversely, some of these pathways can be easily reversed to replenish our citric acid cycle intermediates who might have been removed for other processes as well. The citric acid cycle intermediates can also be used as precursors for amino acid synthesis. Particularly, oxaloacetate can be converted directly into amino acids, in this case aspartic acid, via a transamination reaction, and this process is reversible. Likewise, alpha-ketoglutarate can be directly converted into glutamic acid via the same transamination reaction, and this is reversible. Finally, citric over here is removed to participate in the synthesis of lipids and vice versa. Looking at these three reactions, we can start seeing how the anaplerotic sequences can be used to balance the citric acid cycle. For example, we can imagine a hypothetical scenario where you have a plant cell that's undergoing rapid growth. As part of this rapid growth, you have the synthesis of many carbohydrates. This results in the depletion of oxaloacetate and malate, and these compounds are being converted into PEP or pyruvate via these pathways over here. In order to compensate for this, your plant might start converting its amino acids to alpha-glutarate or its amino acids to oxaloacetate in order to replenish this intermediate and make sure the cycle keeps working, or vice versa. And that's sort of the value or the value of the anaplerotic sequences in maintaining the functioning of your cell, right? Long story short, citric acid cycle, its intermediates can be taken out to be used for various other biosynthetic pathways, which is great. However, that means that these intermediates must also be replenished from somewhere else in the cell, using, again, these same anaplerotic sequences. In this slide here, we're going to start taking a bit of an in-depth look as to these individual anaplerotic sequences. So first we'll look at the anaplerotic sequences in the context of carbohydrate metabolism. As we mentioned earlier, through these anaplerotic sequences, we can either remove citric acid cycle intermediates for gluconeogenesis, or we can replenish citric acid cycle intermediates via carbohydrates. For example, if we're pulling out alpha-ketoglutarate to make a lot of amino acids, we can replenish the cycle directly using carbohydrates. And this can take the form of two different series of reactions. The first series of reactions focus on oxaloacetate and its conversion with either pyruvate or PEP. Conversely, we can also look at the conversion of malate into pyruvate via the malate enzyme. The interconversion between oxaloacetate, pyruvate, and PEP can be summarized as something which we saw earlier in the gluconeogenesis reaction. Shown here on the left is a schematic illustrating the first step of gluconeogenesis. Here we convert pyruvate to oxaloacetate and back to PEP using pyruvate carboxylase as well as PEPCK. These are reactions we saw earlier, and these are the same reactions which partake in the anaplerotic processes. I should point out that these two reactions, pyruvate carboxylase and PEPCK, are irreversible in animals. In other words, these anaplerotic pathways can only really go in one direction, at least in animals. In plants, however, plants have a separate enzyme called PEP carboxylase, which can convert oxaloacetate back into PEP, shown over here. Convert PEP back to oxaloacetate, shown over here in this black arrow. Again, this enzyme is specific for plants. This relationship can be summarized in this scheme over here. This is a summary showing the conversion of pyruvate to oxaloacetate and PEP as occurs in gluconeogenesis. We have the enzymes. Note that this reaction is a one-way street. In plants, we have a separate enzyme which catalyzes the reverse reaction, the reverse reaction here, converting PEP back to oxaloacetate. Note that this reaction is slightly different from the one catalyzed by PEPCK. In PEPCK, we have a ATP cofactor, whereas in PEP carboxylase, there's no ATP at all. We just have the carbonic acid as the sole substrate. Just quickly as well, we can also convert malate into pyruvate using the malic enzyme, and the reaction scheme for this is shown over here below. In this reaction, we simply take pyruvate, combine it with our carboxylic acid and an NADPH to get malate, and this reaction is freely reversible in both plants and animals. The second set of slides starts looking at the conversion of amino acids from our various citric acid cycle intermediates. This occurs via the transamination reaction. In the transamination reaction, we're transferring alpha amino groups to alpha keto acids. And this can be used to directly generate amino acids from either oxaloacetate or alpha ketoglutarate. I should point out that some amino acids can be directly fed into the citric acid cycle at the stage of both succinyl-CoA and fumarate. This input is only specific for certain amino acids, and it is only a one-way street as well. In other words, you can convert isoleucine to fumarate, but converting fumarate to isoleucine is a bit more difficult. And we'll talk about a bit more about that when we start talking about amino acid biosynthesis and amino acid digestion. Taking a closer look at the transamination reaction, what the transamination reaction does is it takes an alpha amino group, shown over here, and it takes an alpha keto acid group, shown over here, and basically switches them. So this amino acid group gets transferred over here, this acid group gets transferred over here, giving rise to the following products. Using the transamination reaction, we can interconvert glutamate and alpha ketoglutarate, as shown over here in the schematic below. This is the structure of glutamate. We have an alpha amino group over here. In other words, an amino group that's on the alpha position to a carboxylic acid. If we do a transamination, this amino group becomes a keto group, giving us alpha ketoglutarate. The same reaction can be used to generate aspartate directly from oxaloacetate. The final anaplerotic pathway or anaplerotic sequence is the synthesis of lipids from our citric acid cycle intermediates, shown over here. This is a relatively straightforward reaction, but one which I figured I would bring to your attention. In order to this anaplerotic pathway stems from a particular problem in fatty acid synthesis. As you can see over here, fatty acid biosynthesis requires acetyl-CoA. However, this acetyl-CoA must be in the cytosol. That's rather inconvenient because acetyl-CoA generated by a pyruvate dehydrogenase in this first step is generated in the mitochondria. Acetyl-CoA cannot actually pass through the mitochondrial membrane, so we need some way of shuttling this acetyl-CoA into the cytoplasm. We can do this by converting it to citrate. So what your body does is it takes your oxaloacetate, it takes acetyl-CoA, and in the first step of the citric acid cycle, via citrate synthase, you make citrate. This citrate is free to pass through the mitochondrial membrane from the mitochondria into the cytosol. Once in the cytosol, the citrate is acted on by citrate lyase, which breaks it down back into oxaloacetate and its acetyl-CoA, which can then go on and participate in fatty acid biosynthesis. Having learned the anaplerotic pathway, we can start applying it to answer some questions. An example of which is shown over here. In this question, we are given a plant cell which is undergoing rapid growth. Under these conditions, the cell requires large amounts concentrations of amino acids for protein synthesis. This question asks what citric acid cycle intermediates would be depleted under these conditions, and what are some ways in which the plant can compensate for this. I'll give you a few minutes to think about this question, and then we'll go over a response. So for the first part over here, what citric acid cycle intermediates would be depleted under these conditions? If our cell is synthesizing large amount of amino acids, we're going to need significant amounts of both aspartic acid and glutamic acid. This results in the release of in the depletion or the consumption of both alpha-ketoglutarate and oxaloacetate via the transamination reaction we talked about earlier. This would be a significant problem because if you start pulling out your intermediate from your citric acid cycle stops working. The plants can get around this by using either the malic enzyme or PEP carboxylase to convert some carbohydrates into either oxaloacetate or malate. This allows the citric acid cycle to keep going, even under conditions where some of its intermediates are being pulled out to make amino acid synthesis. Hopefully this question illustrates the value of the anaplerotic pathways and how the various biosynthetic and catabolic pathways come together, or rather work together to keep your cell working. So the anaplerotic pathways, again, they sort of connect the citric acid cycle with something which is normally considered a catabolic process and connect it to anabolic processes such as amino acid synthesis or carbohydrate biosynthesis. In plants, we can actually take this one step further via something called the glyoxylate cycle. This is discussed in section 13.9 in your textbook. This glyoxylate cycle is referred to as an anabolic variant of the tricarboxylic acid or the citric acid cycle. In other words, it's a version of the citric acid cycle which is extremely specialized for anabolic or biosynthetic pathways alone. In order to unpack what this means, we need to discuss catabolism and anabolism again in the context of the citric acid cycle. As we mentioned earlier, the citric acid cycle is primarily thought of as a catabolic process. It takes acetyl-CoA, breaks it down into carbon dioxide, and releases a lot of energy. In other words, the tricarboxylic acid cycle facilitates catabolism by converting acetyl-CoA into carbon dioxide. However, the tricarboxylic acid cycle or citric acid cycle can also facilitate anabolic processes by providing these intermediates, particularly oxaloacetate and malate, which are important precursors for the synthesis of both amino acids as well as carbohydrates. So that summarizes how the citric acid cycle can participate in both catabolism and anabolism. This leads us to the second question: what steps of the citric acid cycle would we want to bypass in order to facilitate anabolic or biosynthetic pathways and reduce catabolism? We touched touched upon this topic just briefly earlier, right? The catabolic function of the citric acid cycle is due to the fact that it breaks up acetyl-CoA into carbon dioxide. In other words, if we want to eliminate the catabolic properties of the citric acid cycle and focus purely on anabolism and biosynthesis, we need a way to bypass the generation of carbon dioxide, right? These steps in which carbon dioxide is generated represent our catabolic processes. We like to bypass this if possible. If possible. On the flip side, if we want to promote anabolism, anabolism in the citric acid cycle is mediated by these intermediates over here, malate and oxaloacetate. So if we want to focus on anabolism, we want to find a way in which our citrate or isocitrate can be directly converted into malate and oxaloacetate, bypassing the loss of carbon dioxide and focusing exclusively on the generation of these two intermediate species here. And that's what the glyoxylate cycle does. Again, the glyoxylate cycle focuses on converting acetyl-CoA into oxaloacetate and malate, as well as bypassing the decarboxylation process. Shown over here is a schematic of how the glyoxylate cycle does this, or carries this out. Shown over here is a schematic of the citric acid cycle, the normal citric acid cycle. Over here in blue is where the glyoxylate cycle sort of deviates or differs from the citric acid cycle. So in the glyoxylate cycle, we start off with acetyl-CoA, we have oxaloacetate, and that follows the citric acid cycle pathway through citrate synthase and aconitase, ending up with isocitrate. This is where the glyoxylate pathway differs. Instead of decarboxylating isocitrate, it's going to instead carry out a different reaction. This reaction is catalyzed by an enzyme called isocitrate lyase. What isocitrate lyase does is that it takes your isocitrate and cleaves this carbon-carbon bond over here to generate two different species: succinate, which is a 4-carbon fragment, and glyoxylate, which is a 2-carbon fragment, which I've highlighted here in this structure. The succinate 4-carbon fragment is fed directly back into the normal citric acid cycle. Shown over here by this arrow, that's free to react to fumarate, malate, oxaloacetate, etc. The glyoxylate, on the other hand, this 2-carbon fragment, is coupled with a second acetyl-CoA to generate a 4-carbon species called malate via the enzyme malate synthase, which can then go through the citric acid cycle or be used for biosynthetic processes. In other words, the glyoxylate cycle takes the scaffold of our citric acid cycle, bypasses the loss of carbon dioxide, and directly converts your isocitrate into a 2 4-carbon species via a second acetyl-CoA. In other words, it takes the overall scaffold of our citric acid cycle and reconverts it to a anabolic focus. So here I've wrapped this class up with a series of 2 different questions. These are questions which you can think about at home. And that is the end of this class.